DNA damage is linked to basically all of the major diseases of humans. One of the things that we do with the peptides is that we can repair that DNA damage system using a couple of peptides. He was able to significantly reduce mortality. Telomere age was equivalent to a 50-year-old, but 2 years later, with the use of the peptides, his estimated telomere age became 29. And so the goal of our program is to be able to extend people's lifetime.
My name is Warren Thomas. I'm the founder of IHS LLC and the owner of Atlanta Osteoarthritis Center. Today, we have a special guest. Dr. Bill Lawrence is an attorney, former developer, and PhD scientist in longevity. And we're going to touch on a very interesting discussion today. We're going to talk about peptide bioregulators. Not only do they regenerate organs, but they also reduce mortality and even increase your lifespan. Bill, welcome. How are you?
I'm doing just fine. I woke up breathing this morning, so I'm a happy guy.
Any day above ground is a good day, so it's a good day today.
Yes, the same kind of thought.
Today's presentation, again, is brought to you by IHS Wellness, and we look forward to the presentation. Bill, thanks for joining us, and can you kind of give us an indication how you became interested in anti-aging and longevity? Yeah, you have a very interesting story about your family background. Can you go into that really quickly, please?
The short answer is self-preservation. My family history for men going back generations is that they don't live a long time. They all die typically of heart disease, and it happens generally in their mid-50s and early 60s. So my father had his first heart attack at age 57, fatal at 64. And nearly all the other men died in their mid-50s and so forth. And so I knew that history. And I was, doing my entrepreneurial attorney kind of stuff and so forth when my father died. And I decided that I better maybe change my priorities at that point. And so I went back to school and got the PhD. And that was about 30 years or so ago. And I've been working in the longevity field for the last 30 years or so to basically enhance my lifespan to be beyond what my family has experienced.
Fantastic. And you have a concentration in particular with peptides. Matter of fact, specific types of peptides, which are bioregulator peptides and so forth. Can you kind of give us a little insight to what bioregulator peptides are and how you were introduced to them, please?
Peptide bioregulators they're sourced from animals. That is, if we went back in history, 10,000 years, 100,000 years and so forth, medicine was basically derived from plants and from animals. And, 150 years or so ago, when the pharmaceutical industry in America was beginning, all of that kind of got pushed to the wayside, and we went into sort of a chemical process, pharmaceutical drugs and so forth. But there's a long history, of course, of using animals and plants. And in Russia, a well-known scientist, Professor Vladimir Kavinson, looked back into that history, and they determined that they could use amino acids derived from animals to be a powerful intervention for basically organ regeneration. And as we'll talk about when I show some slides and so forth, it started off in the Soviet Union military is where they derived their initial funding from, and the necessity for coming up with something that would be helpful for the Soviet Union military. And what they are, and again, we'll talk about it more when we get into the slides, but basically they're short chain amino acids of 2 and 3 and 4 amino acids in a string, you might call it. And as a result, they penetrate cell membranes quite easily, and they actually act as cellular reprogramming interventions and ultimately for organ regeneration. They can literally regenerate organs.
So Dr. Kavinson reached back into history in regards to peptides.
It's been about 10 or 12 years ago when I met Professor Kavinson in Russia. I was intrigued by the work that the Russians had been doing using peptides for organ regeneration. And so I went to Russia, met with him. He was very gracious to allow me to kind of join them and become affiliated with the institute there. He had many, many studies that he had completed, peer-reviewed studies that he had completed in Russia, but there wasn't anything in America. He was unknown in America. The studies were unknown in America. And so he and I decided that we would do a clinical study here in America using these peptides for organ regeneration. And so we call it the Peptide Bioregulator Protocol. And the goal of this is basically to address aging, longevity, and to use the peptides to change biomarkers that are related to aging. And we're going to get into the detail of that in just a few moments. As an attorney and as a scientist, I'm very focused on evidence and data. There are a lot of claims about various supplements and interventions and so forth, but unless they have data behind them and evidence, then they're really not very reliable. So everything that I do and everything that Professor Kabat-Zinn has done is based on data and evidence. What we all realize, and I've got a slide here that talks about it, where Steve Jobs said that the most precious resource we all have is time, and that's absolutely true, is time, the length of time that we're alive and that we're healthy. And so the goal of our program is to be able to extend people's lifetime. And we have what we call these hallmarks of aging. They're well known throughout the biological science world. And there are about 14 different biological hallmarks of aging. But we picked a couple of them that we think are primary, and that is telomere shortening, and I'll explain what that is, and DNA damage. These are vital intervention points for creating longevity in a human being. And, a lot of really very well-known scientists have concluded the same thing. This slide talks about Dr. Venki Ramakrishnan, where he's written a book that's titled Why We Die. And what he's saying is that as we age, the cell's quality control machinery deteriorates. And let me explain what that is. In our cells, we have this repair mechanism that when a cell's replicating, because life is all about cell replication, as cells replicate, they have to replicate perfectly in order to keep the organism alive. And as we age, that repair system begins to kind of wear down, and it doesn't work as perfectly as it did when we were younger. And so one of the things that we've done with these peptides is we can use these peptides to basically rebuild the DNA repair system, and that allows for significantly additional cell replication. And as I said, several other scientists have come to the same conclusion. This is Jan Hoschemaker, who is saying that the aging hallmarks are secondary consequences. It's the accumulated DNA damage that contributes to systemic aging. And another scientist, Jan Vig, has talked about, based on the abundance of evidence, DNA damage is now considered as the single most important driver of the degenerative processes that collectively cause aging. And we know that DNA damage is linked to basically all the major diseases of humans, like cancer, to diabetes, and so forth. In other words, if your DNA repair system is working as it should and as it was when we were younger, then all of these diseases tend to be minimized. It's as we age and that system breaks down that we then become vulnerable to these kinds of problems. And one of the things that we do with the peptides is that we can repair that DNA damaged system. So Professor Kabat-Zinn and I agreed that we would run two clinical studies. One is a telomere study and the other is a DNA methylation or epigenetic study, and I'll get into the detail of how they work. And the peptide bioregulator longevity protocol, as we call it, started in 2017. It basically kind of finished up this year, well, actually last year, 2025. It's a scientifically proven, based on published clinical human studies, and American laboratory data confirms the following: mortality reduction, organ regeneration, and biological age reversal. And today I'm going to show you the outcome of each one of those. And what we found, Professor Kabat-Zinn's work basically showed that the peptides could increase mean lifespan by about 20 to 40%. And we do this by activating telomerase and lengthening telomeres, as well as organ regeneration. So mortality reduction, let's talk about that. Professor Cavinson, literally 20 some odd years ago, was doing clinical studies where he was able to significantly reduce mortality using a couple of peptides. Specifically, he was using the peptides for the pineal gland and the thymus, and there are 21 of these peptides now. He was using 2 of these to significantly reduce mortality. And he has this is a published clinical study that I'm showing here, where he was able to reduce mortality, and I have a graph of it here to explain to people. What this graph is showing at this point is that they had a control group of people. These people were 60 to 74 years of age. And if we look at the mortality rate in the course of 12 years in this study, the people in the control group, that is the non-peptide people, 44% of those people died over that 12-year period of time. In the group that was taking just one peptide, remember there's 21 of these now, the group taking one peptide, the pineal gland peptide, the mortality rate was 22%. In other words, it dropped by half taking just one peptide. That was how powerful the pineal gland peptide is. And then a couple of years later, he developed a second peptide for the thymus. In looking at a group of people, what we call the old people, the Russians are not the most diplomatic as such. In this group of 75 to 89-year-old people, in the control group, the non-peptide group, 80% of these people died. 80.2% of these people died over a 6-year period of time. In the group using just the one peptide, the pineal gland peptide in this age group, the mortality rate was dropped down to about 46%. When they added the second peptide, the mortality rate was 33%. So we went from basically about 82% mortality down to 33% over a 6-year period of time using just 2 peptides. And Warren, as I mentioned, we now have 21 of these peptides. And then the next thing that the peptides can do is organ regeneration. And what we're showing with this slide is that the Russians at the institute that I'm affiliated with treat various retinal issues using these peptides very successfully. And I'm showing some slides here using, in the case of diabetic retinopathy, we have over on the left here, where you see all of the discolored, this is what we call an eye scan, a field of vision eye scan. Where you see some green, that's where people have vision, but where you see all the red and the black and the yellow, that's impaired vision. 2 years later, after the use of peptides, we have in about 85 to 90% of the scan is green. In other words, people have had vision restored at this point. And this is true in the various diseases of the retina. So for instance, this is macular degeneration, the same kind of thing where you see a fair amount of black and yellow and so forth in the before. And then after a couple of years of treatment, you see a lot more green. So their vision has been improved. And the same thing with retinitis pigmentosa, where you see the black, this person basically would be very impaired in terms of their vision. And after treatment of about 2 years, there's a significant amount of green. Unfortunately, here in the US, there is very little that can be done for these vision issues, for these retinal issues. They have some drops that maybe slow the process, the degeneration a little bit. But in Russia, at the Institute, they can restore a significant amount of vision using the peptides. So, as I say, we use these peptides for a lot of things, including cancer. They're used to reduce tumor incidence. And a fellow by the name of David Sinclair at Harvard has said, I'll read his statement here, "One day it may be possible to reprogram cells by pills." Well, the Russians have already done this with pills because the peptides that we're using in our clinical studies are oral capsules. They come in in an injectable form as well, but in our study, we use only the oral capsules. And because they're just 2 and 3 and 4 amino acid strings, They pass through all the membranes, including the blood-brain barrier and so forth, intact, and can be used as cellular reprogramming agents that results finally in organ regeneration. And so the peptides activate genes for cellular regeneration. We're going to talk about telomeres. We're going to talk about epigenetic restoration and so forth. And so what we did was we created a clinical study using these peptides, and we have about 130 or 140 people in the clinical study. The peptides come from animals, as I mentioned earlier. They're isolated, purified, and fractionated into amino acids, and then created in an injectable form or in a capsule form. And where this started from was back in the 1970s. The Soviet military was aware that the United States was developing lasers and other devices that would have damaged vision, damaged hearing, and so forth on the battlefield. And the Soviet Union people were doing the same thing. And they said to scientists, including Professor Kavinson, they said, you need to come up with some way of restoring vision, restoring hearing, and so forth, if in fact these lasers become effective. And so they put together a group of doctors and Kavinson and other scientists and so forth. And they then developed these peptides based on basically using these animal-sourced amino acids. And so then they spread the use of these peptides to the military because they were realizing that when they sent out submarine personnel out on these extended trips, that when they came back, that because they were sitting in submarines with pretty primitive nuclear protection, that they were having all sorts of thymus problems and other issues and so forth. And so they started using these peptides to restore the thymus to begin with. Then they used them for the cosmonauts, and they used them eventually for Olympic athletes in Russia. And these studies then with the peptides weren't just restricted to the Soviet Union and Russia. They actually partnered with the National Institute of Aging here in the US, with the Italian National Research Center, with German institutions, Spain institutions, and all of these were involved in the development of these peptides. So it wasn't just a Russian project. And as I said, there are now 21 of these peptides. There's a peptide for the brain, the heart, the bladder, pineal gland, for muscle, for the eyes, for the liver, kidney, et cetera, et cetera. And there's a lot of discussion these days with regard to peptides, and particularly what we call the synthetic peptides. And so I need to explain the difference, Warren, between the synthetic peptides and the peptide bioregulators, because they're actually very different. The synthetic peptides that are very popular today, that are done with injections, are basically used for acute conditions like wounds, ligaments, stomach issues, etc. And they are injectable peptides, and they are based on chemical composition. The bioregulators are different than that in that they are sourced from animals. And they are a longer term kind of intervention. That is, it takes several years to see the benefits of the peptide bioregulators, because what they're doing is regenerating organs. So we used them in this clinical study, reverse biological age, and we used what we call the telomeres, and we'll get into the epigenetics. Those are two primary biomarkers for biological age assessment and for basically creating longevity. And so we decided that we would use the peptides to see if we could improve these biomarkers. And telomeres are the end tips on the end of our chromosomes. And every time there's a cell replication, we lose a little bit of this end cap. People sometimes will use the metaphor of the little plastic tips on the end of sport shoes and so forth. Every time that you lace up that shoe, then you lose a little fraction of that little plastic end cap. Well, the same thing happens with our cells, that as cells replicate, we lose a little bit of that. And what has been found scientifically is that when you have lengthier telomeres, you have better cell replication over time, as we age and that little end cap begins to kind of fray, you might say, then the replication is not as perfect as it was when you had longer telomeres. So telomeres are a good biomarker for determining longevity. And we wanted to know, if we use these peptides, could we actually restore telomere loss? Could we lengthen telomeres? And a number of American scientists talk about telomeres. Here we have Bill Andrews saying every time our cells divide, our telomeres get a little shorter, and every time they shorten, our cells age. And then we have Elizabeth Blackburn, a recipient of the Nobel Prize in 2009, says, that telomere slows the rate of which telomeres degrade, and research indicates people with longer telomeres have less risk of developing the common illnesses of aging. And then another fellow, Michael Fossel, scientist, has basically said that telomerase activation is the single most promising approach to reversing the effects of aging. I have great respect for Dr. Fossel, but I think he's overstated it a little bit, because we have also what we're going to get into, epigenetics, that are as important, if not more important, than telomerase activation. And in our clinical studies that we're running here, we address both telomeres as well as epigenetics, and we'll get into that in a few minutes. So people with short telomeres generally are very vulnerable to most of the aging problems that we have, cardiovascular disease, macular degeneration. So people with longer telomeres have less vulnerability to these things. People with short telomeres are more vulnerable to them. So our clinical study involved 124 participants. They're primarily physicians. We didn't ask people to change anything else in their lifestyle and so forth. The only thing that was different was that they were using some of these peptide bioregulators over a period of about 3 to 4 years. And these are some slides from SpectraCell Labs showing a significant increase in people's telomeres if they've been on the peptides for a couple of years. And so we have one individual here whose age was about 72, I think, his chronological age. And he had really good telomere age at baseline. His telomere age was equivalent to a 50-year-old. But 2 years later, with the use of the peptides, his estimated telomere age became 29. In other words, we were able to lengthen his telomeres to that of a 29-year-old. And then a few years ago, an Olympic doctor contacted me because apparently they do extensive testing of the Olympic athletes and so forth. And to my surprise, one of the tests that they do is a telomere test. And so the doctor, this is Tora Bright, Olympic gold and silver winner in skiing and so forth. And she was at the time 32 chronologically. She had won the Olympic gold and so forth. But when she was tested for telomeres, her telomeres were equivalent to a 56-year-old. Now, the reason for that is that what we know from a lot of studies is that the number one situation where telomeres are accelerated in terms of the loss of the length is under stress. Stress is very damaging to telomeres. And so with Tora Bright, at age 32, her telomeres were equivalent to a 56-year-old. And of course, the reason for that is that she had been a competitive skier since she was a young girl. And so she had this huge loss of telomeres based on the amount of stress. So the doctor contacted me. We put her on a program. And I think it was about 18 months later, her telomeres were restored. They were lengthened, and they were equivalent to a 31-year-old person. And then these are just various other people. This is Vess Jones, and she happens to be my wife. Her baseline test when she was 60 years of age, her telomere age was 63. In other words, her telomeres had been accelerated loss by a couple of years. This is very typical of what we see with Americans. They're typically, what we call telomere age is a couple of years older than their chronological age. And I think a lot of that has to do with the fact that we live in a high stress kind of environment here in the States. So we put her on a program, and I think about 2 years later, actually 3 years later, she was 62 2 years later, and we lengthened her telomeres equivalent to a 33-year-old using the peptides. And these are my telomere results over a period of time. In 2014, I didn't know anything about the Russians and Kabat-Zinn and the Institute and so forth, and I knew very little about telomeres. And so I did a telomere test, though, among many other tests that I was doing at that time on myself. I was 68 at that time. My telomeres came back equivalent to a 75-year-old. In other words, I had accelerated telomere loss at this point. And I had been under a lot of stress. I was in my entrepreneurial period, Warren, at that time, and I was building shopping centers and medical buildings and so forth. And it was a high stress period of time. And so that was my situation where I had accelerated telomere loss. That does not bode well for a long life. In 2016, well, actually in 2015 is when I discovered what the Russians were doing. I went to Russia, met with Kabat-Zinn, became affiliated with the Institute at that point, got onto a peptide program. And so in 2016, I retested, and my telomeres at age 70 were equal to a 68-year-old. Then 2 years later, continuing on with the peptides and so forth, at age 72, my telomeres were equivalent to a 44-year-old. And I continued on, and eventually, the most recent tests that I have at age 76, my telomere age was equivalent to a 23-year-old. As a result of taking these peptides over a period of a number of years. Now, what does that mean when I say, my telomeres are equivalent to a 23-year-old? What that means is that I will have additional cell replication, because the telomeres are what allows for cell replication to occur. I'll have additional cell replication, which, barring an accident or some other disease, cancer and so forth, means that I should have a fairly long life, because I'm going to have more cell replication than a person who has shorter telomeres. So this is a spreadsheet that I'm showing here. As I say, about 100 plus people in the telomere study. What this shows are the results for all of these people. And what we have is a pretty big range in terms of what their telomere results were. We have some people who got, over a period of several years, a 30-year increase in their telomeres. We have other people that only got a year or two. And the difference between them primarily is the amount of stress in their life, because I mentioned earlier that stress is just really very impactful on telomeres. But the average decrease in the what we call cellular or biological age or telomere age was 21.62 years over a 3-year period. In other words, using the peptides, we were able to lengthen people's telomeres to the equivalent of almost 22 years at this point. And that should give them pretty significant, that's 2 decades of additional potential longevity. So then in 2000, about 5 years ago, we decided to add epigenetic study to what we were doing, and I'll explain what that is in a moment. Epigenetics is all about what genes are turned on and off in our system. And things that impact genes being turned on or activated or turned off are things like diet, stress, exercise, sleep, nutraceuticals, peptides, environment, et cetera, et cetera. And so every cell in our body has this DNA sequence. And what happens is that the body can basically have what we call a DNA expression can be turned on and off. In other words, genes that are related to longevity can be turned or turned off, impacted by things that I just mentioned, like sleep and stress and so forth. And so we knew that the peptides could turn on genes that are related to longevity. And so we set up this study. And Professor Kabat-Zinn had published a clinical study where he determined that the peptides are capable of complementary binding to certain short nucleotide sequences. In other words, can impact DNA. And he wrote a book about it called Peptides in the Epigenetic Control of Aging. And what happens is that these can actually turn back on the longevity genes. And a couple of years ago, it became possible to actually do a test to determine which genes were turned on and to the extent. And Dr. Stephen Horvath, UCLA, was able to figure out that if you're what we call your epigenetic age, if it's turned on and it's older than your chronological age, that you have an increased vulnerability to aging and to all sorts of diseases and so forth. What he found was that if you're aging 8 or more years faster than your calendar age genetically, that equates to twice the typical risk of dying. While if you're aging 7 years slower, it's associated with half the risk of death. In other words, we can measure this at this point. And so what these charts show is an increased mortality risk. If your epigenetic age, as we call it, is 8 years faster than your chronological age, you have a 100% increased mortality risk compared to your same age peers. At the same time, you could have a decreased mortality risk if your epigenetic age, as we call it, is less than your chronological age. Then, for instance, at seven years less, you have a 50% less vulnerability compared to your same age peers. And so what we did was use the peptides for this epigenetic testing. And what we're showing with these different slides here is that people who had baseline, like this fellow here, his DNA age, as we call it, was 55, his calendar age was 53. And what that means is that his genetic age was 2 years older than his calendar age. And that represents about a 25% increase in vulnerability of all-cause mortality. So we put him on a peptide program, and 2 years later, he was 55 at this point, but his DNA age or epigenetic age was 51, 4 years less. And at 4 years less, he has about a 35% decrease in his all-cause mortality risk. So in my own case, that in 2020, I had been on the peptides for about 5 years or so at that point. And my baseline test, because this epigenetic pace of aging just became available in 2020. So my pace of aging at that point was 0.92. In other words, I was aging each year 8% slower than my chronological age. Then in 2021, with the peptides, I was able to slow that down even further to where I was 9% slower than my chronological. In 2022, I was 11% slower. In 2024, I was actually 21% slower than my chronological aging. What I was saying is that the technology has now gotten to the point where not only can we determine an epigenetic biological age based on epigenetics, not only can we determine an overall epigenetic age for a person, but we can actually go in and make a determination of various organs and how old they are biologically. And so this slide here is showing, for instance, we have the lungs, we have the blood, the liver, we have kidney, the heart, the brain, and so forth. And we have a epigenetic age estimate for each one of those. And so now when I create a protocol using the peptides, I can address the ones that have accelerated aging. And what we have found over the period of the last few years, that with the peptide program over a couple of years, that we can improve the biological age or reduce the biological age of all of these individual organs. And this slide is showing one of those samples where you see the green arrows. Every one of these over a 2-year period of time improved in terms of the specific organs. Okay, so based on the epigenetic study, now I've talked about the telomere study, now we're in the epigenetic study. What we've discovered was over a period of a couple of years that we were able to reduce people's epigenetic age or the biological age based on epigenetics by almost 5 years, 4.67 years. Now, some people will say, well, now you were able to lengthen people's telomeres by 22 years, but epigenetically, you only reduced it by about 5 years. The difference is that we don't look at epigenetics in terms of the number of years. We look at the percentage of all-cause mortality reduction. And a 4.67 reduction in epigenetic age is almost a 50%, about actually specifically a 56% risk of all-cause mortality. So in other words, by using the peptides epigenetically, we can decrease a person's risk of mortality by more than 50%. So the result of both of these clinical studies, the telomere study, using the peptides, we're able to increase people's telomeres, and the equivalent would be about a 22-year increase in their longevity. And in terms of the epigenetics, about a 50 to 56% reduction in all-cause mortality risk. So the two were very successful studies. And this is what I call the bridge, the longevity bridge. The peptide bioregulators as powerful as they aren't the end all, because the way I look at it is that the peptides are an interim thing that will get us organ regeneration, extended life, less vulnerability to disease and so forth. But we're on the cutting edge now, and this is what I call the bridge. We're on the cutting edge right now. If you can stay very healthy for the next 10 years, the things that are on the drawing board that are being produced right now, like artificial intelligence and things, will allow us to leap forward by probably 10 or 15 more years. And so the idea with the bridge, Warren, is you stay as healthy as you can for the next 10 years, because then you can leverage that into probably another 15 or 20 years beyond that, based on the things that will be available. And some of those things, are being funded by billionaires at this point. They're bankrolling research and so forth in terms of longevity. And there are things like quantum computers, replacement organs, robotics, nanotechnology, 3D printing, and so forth that are on the drawing board. Some of these actually in place now, artificial intelligence. And a huge amount of money is being spent at this point on that research in order to basically look at extending people's lives very significantly. And this slide here shows an interesting metaphor here. This is a slide of a man who has a 1966 Volvo that he drove more than 3 million miles. And he did that by constantly maintaining the vehicle. He did all the work himself and so forth. Unfortunately, if you see the picture, he's kind of overweight and graying and so forth. And unfortunately, he did a better job of maintaining his vehicle than he did of himself, because he died a couple of years ago at the age of, I think it was 76. So this is the current aging expectation that everyone has. We're young and we're healthy and so forth. We get to middle age and then we get into our senior years and we become frail and so forth. What we have in using these peptides is that we're able to take that situation and regenerate organs so that the person ends up being healthy again. And that's basically the program, Warren, that we've created. The clinical studies, very successful in creating the potential for significant longevity enhancement.
Okay, fantastic. Bill, a couple of questions. The participants in the study that you completed recently, they were primarily healthcare professionals, am I correct?
Yeah, primarily physicians, chiropractors, health professionals. Yes, I would say about 2/3 of them were.
And what was the feedback from the participants themselves in their results anecdotally?
That's hard to gauge because, if you're doing an organ regeneration, you're not going to necessarily feel anything particular. That's why the lab tests are critical. But everybody basically, when we originally set up the clinical studies, we thought they would run about 3 to 4 years. And what's happened, we figured that, after 3 or 4 years, we would reach the goals that we set for people, because we actually individually set telomere tests, goals and epigenetic tests, and we most of the time reach those goals. So for instance, say a person who is 70 years of age, we would want to reset their telomeres to that of a 40-year-old, and we can basically do that. Epigenetically, we want people to be 6 or 7 years less than their chronological age. So we assume that people would get that point, and we have been successful in doing that, and then they would leave. Problem is, nobody wants to leave. They all want to stay on a maintenance program. And so the reports we get is that they don't get sick very often. They have good energy and so forth, but it's not a dramatic kind of thing. It's over a period of years, Warren.
And that's very important for the audience to understand that damage to the human body, may take time. Healing and regeneration will also take time. So someone engaged in this protocol, I think based on our conversations, they would have to understand that this is not an overnight fix. Am I correct?
Very correct. In fact, we tell people, and we actually have them sign a disclosure document. It's not a contract, but a disclosure document where we explain to them that this is a 3 to 4 year process. It takes that long to get the telomeres and to get the epigenetic age to where we want it. And so they have to have patience, because we're talking about rebuilding, an organ, and it's not a fast process.
And I think in the introduction, you mentioned that Dr. Cabrinson was affiliated with different organizations in different countries. So this is not studies limited to Russia, but including what you've done recently, you have other participants in other countries supporting the evidence of the benefits of the peptides, the bioregulators.
Yeah, that's very correct. There were about 5 different countries, Spain, Germany, the US, and so forth, that were involved in the studies over a period of time. And of course, my study, or I should say our study, has been going on for 7 years here in America.
Fantastic. And in a previous conversation, you had mentioned anecdotally a story regarding retinitis pigmentosa in 2 young men. As I've mentioned to you before, I have retinitis pigmentosa, and also one of my siblings has it also. And we found that it was not necessarily hereditary. We have not been able to identify a few generations back that other individuals within the family line have had it, but somehow myself and my younger brother, we do have retinitis pigmentosa. Can you speak to the anecdotal information about the 2 young men that you assisted and have seen good results with retinitis pigmentosa?
Yeah, the situation was very early on. I think this would be about 2016 after I was introduced to the peptides and had been working with Professor Cavinson for a year or so. A friend contacted me who had some friends where the 2 brothers in this family, they were teenagers, I think around 13 and 16 years of age. Within the same 12 months, both of the boys had been diagnosed with retinitis pigmentosa in, early stages at this point. And so the friend contacted me and asked, he was aware that I was doing something in Russia with peptides and so forth. So he asked if there were peptides that might be helpful with their vision. And I explained to him, I'm not a doctor, so I can't be treating, but there are peptides for the retina, vision peptides we call them, that maybe would be helpful. I can't promise the outcome and so forth, but we would give it a try. And so the parents were willing. We went to their eye doctor, the ophthalmologist, and explained what we had, these peptides. And the ophthalmologist was very open to it because the peptides are natural elements. They're supplements, they're food supplements directly obtained from animals. And so they're not chemicals. So the ophthalmologist was willing to give it a try. So we put them on the peptide program, and we always use a couple of different peptides. So for vision, we would use the vision peptide, we would use the arterial peptide, we would use the thymus peptide for general immune support. And over the first year, there wasn't any degeneration whatsoever in their situation. In fact, their night vision improved within about 6 months. 10 years later, they have perfect vision at this point. They can drive a car, they have jobs, they have families, and so forth. And so we assume that the peptides probably had a lot to do with retaining, because normally with retinitis pigmentosa, it's a progressive disease. And over a period of a couple of years, people tend to lose a lot of their vision.
Exactly. I'm a prime example of that. In regards to the biomarker, you mentioned that outside of the epigenetic, you also do the telomere biomarkers. For our audience, is it important if they're going on a protocol that they get tested for those biomarkers as part of that process?
Oh, yeah. Our baseline testing before we start the protocol at all is to measure their telomeres. Different labs measure them, as well as getting epigenetic labs measured so that we have those as baseline biomarkers so we can compare and track the improvement.
Okay.
Then, Warren, we retest people every 12 or 15 months.
Exactly, to see that progression. Now, you had mentioned earlier 2 types of peptides, the peptide bioregulators and the synthetic peptides.
Yes.
The United States, there's been a growing movement for the encouragement and use of peptides. And matter of fact, we see it in the weight loss space. Peptides such as semaglutide, the pharmaceutical name Ozempic or Wegovy, tirzepatide, and now we have one coming up called retatrutide and so forth. These are all peptides, but they're chemical-based. For those individuals who are currently utilizing the chemical-based peptides, can they still utilize the bioregulators bioregulator peptides also at the same time?
Yes, they can, Warren, because they're doing two different things. The synthetic peptides are what I would call used for acute situations like weight loss and so forth. The BPC 157 is used for wounds and ligament restoration and so forth. The peptide bioregulators are focused on organs and are focused on regenerating organs. So they're completely two different tracks, and there's no conflict between them. I have a number of the doctors using the synthetic peptides as well as the peptide bioregulators, and I think they're a great combination to be using both.
I think so. You're targeting several things at the same time, giving the body support in relationship to regeneration. So it makes a lot of sense to have both types of peptides in combination. So I think this is a lot of very interesting information, Bill, and I would love to have a future conversation with you and kind of dive into some of the specifics in regards to telomeres and organ regeneration. And I'd love to have you back part of that discussion. For our audience who may be interested in learning more, and eventually, if they're interested in learning more about the protocols, I know that Bill tends to limit the number of inquiries, and what we're offering is an opportunity that if there is an interest, reach out to us. Contact information will be somewhere in the video, and we can then share that information with Bill, that if you have a need, in particular for anti-aging and longevity, Bill can review and see if it's something that he can support and assist with. Am I correct in that assertion, Bill?
Well, yes and no, Warren. I have a private life, and I'm very guarded about keeping it in balance and so forth. And so I don't ever give out my contact information, generally, even when I speak at a lot of medical conferences and my information is not given out. I make it purposely difficult to reach me because it's a screening process. If someone is really passionate and serious about their longevity and their health and so forth, then they've got to work to find me a little bit. And what I do is people like yourself, podcasters and interviewers and so forth, you're my screeners for me. If you have someone that contacts you who's very interested, very passionate, and there's fees involved, there's costs involved in doing all of this, then you can pass that information to me and let me know something about them and so forth, and then I'll let you know whether or not I'm willing to have contact. But I don't want anybody contacting me directly. I just request that you don't give out my contact information.
Wanted the audience to hear it directly from you because that's something we've discussed before. And folks, like I said, we're here to share information through our IHS Wellness and Atlanta Osteoarthritis Center. And as Bill said, if you're passionate about anti-aging and longevity, you can reach out to us and we can share that information with Bill. And if Bill sees that there is some passion behind it, and most importantly, understanding that there is a cost to this, we definitely would see what can happen for you and so forth. But Bill, again, thank you. Looking forward to speaking with you soon in regards to peptides and how this industry is going to grow and all the opportunities that are going to be created via that bridge you had mentioned for good health and longevity. Again, thank you.
Warren, it's been a pleasure. Thank you too.
I believe, like Bill Lawrence, that the next 10 years, if you are able to maintain your health, you're going to see tremendous changes. And I believe many of us will have lifespans that may exceed 90, 100, 110 years, but not only longevity, but longevity, sound body and mind, and good health. So with that, if you'd like more information, click in the description, reach out to us, schedule a consultation. And if you're in the Atlanta area, you can also schedule an appointment. Thank you. If you're enjoying the video, please like, subscribe, and leave a comment. The algorithm loves that. This was an interesting topic. If you enjoyed this video, here you can click on the next one.